So, hello everyone and happy, healthy new year. Welcome to this new season of the BioClock Academy lecture series. We hope that we can entrain you also in this year with some enlightening talks, which can give a boost to your clocks and help you get more insights into this amazing research world. Happy to see many familiar faces, but also new ones. My name is Juana Georgiana Rus-Oswald. I'm a postdoctoral researcher in the BioClock Consortium, and together with Hannah Brooks, which is a PhD student in the same consortium, we will host the session of today. For the ones of you who are new and the first time in this environment, the BioClock Consortium is a Dutch research organization which was set up to study different aspects of the biological clock in health, in society, and in nature. And the aim of this lecture series is to introduce you to the basic concepts and equip you with the proper knowledge from experts in the field. And the lecture series takes place every third Wednesday of the month. And for the time frame of today, we will have like 40 minutes talk followed by 15 minutes of discussion. So roughly 1 hour so that we can close at 5 Central European time. And during the talk, you can put your questions or write your questions in the chat and we can discuss them at the end, but it will be also open mic at the end. And now I give the floor to Hannah, who will introduce you to our guest speaker today.
Yes, so also Happy New Year from me. Today we have kind of a special edition because we will feature one of our internal BioClock researchers, Dr. Stephan Michel, who will actually retire from his position as assistant professor at the LUMC in a few days. Luckily for us, he will stay active within BioClock a little bit longer, but still we thought this is a nice opportunity to highlight his work. So over his career, Stephan has really worked on a diversity of topics and also over a wide range of study organisms, including scorpions, mollusks, mammals. And since 2004, he's active in the group of Johanna Meyer, where his work is mainly focused on the mechanisms of synchronization in clock networks and also the effects of aging on biological clocks. And he gave today's talk a very poetic title, Old Clocks in Modern Times. So he will tell us about what problems, challenges older clocks face in our modern society, how this is studied, and how we can possibly solve the problem. So please, Stephan, the floor is yours.
Okay, well, good afternoon, everyone. I'm going to share my screen now. Let's see if it works. Okay, so good afternoon and also Happy New Year to you from me. And if there's anything wrong with the audio, so I'm going to hear it from Georgina or Hannah. Thank you, Hannah, for the nice introduction. That was very kind. You know, everybody asked me, well, you're still around here, what's going on? So I have another 2 days in the lab, and then I'm going to do other things, but also continue to work a little bit on clocks. So what I thought about today was kind of fitting, of course, you know, it's old clocks in modern times. And I actually was, I think Jörg Meyer was suggesting this title a while ago when we gave a talk at the sleep meeting. So it's not completely for me, you know. Anyway, so let me just get a pointer here. And what I'm going to do is to introduce you very briefly. A lot of people know, maybe some not. You know, we have a central circadian clock in the hypothalamus. This is the suprachiasmatic nucleus. It's called that way because this is where the optic nerves cross, and light is received by the retina, and by special cells. This information is conducted to the SCN, and we actually this clock needs this light to entrain or to synchronize to the outside environment every day. Now, importantly, this central clock is organizing, or is a temporal order in the body, in the whole body, not only in the brain, and which results in changes in physiology over the time of the day, like body temperatures dropping in the night, blood pressure, growth hormones, and many other things. And of course, because of physiology and behavior are regulated that way, it's no surprise that also diseases are following this kind of circadian modulation, and the circadian clock actually plays a lot of role in keeping the body healthy, which is kind of an interesting, twist to the whole story in my regard. Now, don't forget, we long thought about the SCN as a master clock, a master-slave idea and so on. But in the meantime, I would rather call it a central clock, which is organizing things more like a conductor. I think Mike Menaker suggested that originally in the church. And to conduct an orchestra and, the different instruments being then the different peripheral clocks and organs and tissue, and that, the organ, if the instruments play at the right time, then, the whole symphony sounds right. And if not, then we have a problem. But also, this instrument sends signal back to the conductor in terms of, when should I be ready, or I'm ready now, or I have enough, slept enough, that kind of stuff. So this is important, and we understand now that we shouldn't neglect this kind of feedback. Now, if everything goes well, then and we have no challenges from the outside, then we have a perfect clock that runs about 24 hours and is reset every day by light. And we have the internal rhythms are actually reinforcing this clock. And we have, hormone rhythms at the right time, the sleep and arousal at the right time, and regularly the body temperature feeding rhythm, exercise, and social interaction. So this is all supporting, every rhythmic input is supporting the synchrony within the system. But we have, of course, and this is only one influence. We have, of course, alarm clocks. We have a lot of light at night. Some of the BioClock consortium are looking at light pollution here. So we have a lot of influence, and that's only part of it. We're eating not at the right time, and all that is a challenge to the internal clock. And this is then where it goes wrong. You have artificial light going in, or you have altered hormone profiles because of stress or shift work. You have sleep disruptions for whatever reasons. You eat at the wrong time, or different high sugar or high fat or sugar, and you don't move too much. You also have a sedentary lifestyle. So this all leads to problems. And in a review a few years ago from Takahashi lab, we saw he noticed a few things like sleep disorders, like this is a genetic disorders, or metabolism. And I think there was already a BioClock Academy lecture on that one, where it can be a big problem for obesity and diabetes. The microbiota in our guts also have a clock and communicating actually with the rest of the body, and this is also important to not be dysregulated. And the consequences of course, in cardiovascular, I think Frank Scher maybe told about this, is also very important. And finally, even cancer seems to be a link between clock disturbances and the enhanced proliferation. But what I want to talk about today is a little bit the effect of aging. So we know that in age is the damped amplitude of the rhythm, and we actually have the stress response is increased. Now, why is aging such a problem? Well, unfortunately, we have an aging society. So a lot of Western societies are actually have a reverse population dynamic. So we have here the pyramid, which is the usual. That's Netherlands in the 50s, and this is Netherlands in 2023. You already see that there's a lot. In my age here, there's a growing number of people. So with this growing number of older elderlies in the society, we have all the risk factors increase for different diseases. And the elderly often have sleep disorders that are partially based on clock dysfunction. And the broken clocks also accelerate the progression of diseases, which makes it even worse. Now, if we identify interventions to restore these clock functions, that could be beneficial to different diseases, but also increase the quality of life. In a nutshell, and it's just an outlook, and I'm going to pick a few things to talk about. This is a review from us, from Renat and myself, a few years ago, where we laid out the importance to look at the different levels of analysis. So we have, of course, this is just the FCN clock, and this, as I said before, this is very reductionist now, and there's lots of more, and we are going to mention that at the end. So we have the input, either light, food, activity, that goes into this clock, and then we have different layers, like an onion, a bit, and you have the molecular clock, you have the cytosolic oscillators, membrane components, and neuronal networks. So more than one neuron. So in the cell, you have the molecular clock that we found is less affected. We have a calcium rhythm that is affected. We have membrane, if you go to ion channels that are really affected by age. So the more you go into the cell, it's actually the cellular level is very affected. Whereas the network is actually still managing to, produce a rhythm with a reduced amplitude. So this is sort of the brief outline what I'm going to talk about. So the hallmarks of age clock, and this is from a lab. This is actually in vivo recording from mice in the SCN in this central clock, and this is electrical activity, which beautifully goes up. This is the highest during the day. This is light. This is dark, and low during the night. And this is the behavioral activity. So this is a night active animal. And if you switch off the light, then it actually continues with a slightly lower amplitude, but it actually continues beautifully. And this is from a lab, actually from my postdoc advisor, Jean Block and Christopher Colwell, where I also worked for 4 years in LA. So that showed that they actually have a reduced amplitude. The about 16, 17 months old mice, about 50% reduced amplitude. And you see here a very disturbed rhythm here. So what we wanted to know is what's the basis of this reduced amplitude, and for that we actually also applied our ex vivo techniques to record in slices. And the idea was what we had from previous work from 2007 on photoperiod is that there can be a reduction in amplitude if the single cell rhythms actually are more distributed in phase. So you can imagine if they're very synchronized and very close together, they produce, as an ensemble, a very high amplitude. And the width will also be shorter, which is important here in this case for encoding day length. And so during the seasons, this happens and changes, but what happens in aging? So we looked in the old, in slices and brain slices from old animals, old mice, and these are like 24 months old mice. And you see again also in ex vivo, so in the slice, this amplitude is reduced by about half. And if you look at the single unit activity, it's very much distributed throughout the day, whereas the young one is more synchronized during the middle of the day. And this is a summary here. And you see that in the young, you have a main peak during the day. And in the old, you actually have two peaks, which are 180 degrees out of phase. So it's sort of not completely disorganized, but there is sort of a second population of neurons that fire during the night. Now, what could be behind that and what is the basis of that? We don't know yet how this phase distribution is exactly established. But one of the things that is a very important feature of networks is the homeostasis of a balance in excitatory and inhibitory activity. So you always have to have a balance of neuronal activity of excitatory and inhibitory. And the normal one, like in the cortex, about 70% excitatory, 30% inhibitory. You know when that goes wrong, for instance, in neurodevelopment, where you have not enough inhibitory synapses developing, and you get kids that develop epilepsy, and you have to treat them, and it's really a serious disease. A number of serious diseases can happen. So in aging, we already know that also in the cortex, this inhibitory number of inhibitory activity is decreasing, and we wanted to know, is that also the case in the SCN? Now, in the SCN, we have not the classical situation where we have like glutamate as excitatory, GABA as inhibition, because we have mainly GABA, but in the SCN, it can be excitatory and inhibitory GABA. So now that makes our goal, our task a bit easier, that we can actually use calcium imaging. So these are slices stained with certain with Fura2, which is an indicator for calcium, intracellular calcium. And by imaging them, we can actually determine for single neurons the reaction to an application of an agonist like GABA. So we want to know, do these cells, or which cells react as excitatory, as seen here, and which cells react inhibitory, as seen here in the calcium response. And then the ratio between excitatory and inhibitory, in this case, we define as the excitatory to inhibitory balance. And we found that indeed in the number of inhibitory responses decreases, which leads to an increase in the EI ratio. So you have a higher EI ratio in older SCN. And while that could contribute, so if the EI balance is higher, so higher EI balance correlated to wider phase distribution, if you remember that from the photo period, what I just said. So the change in EI balance may contribute to the reduction in synchrony within the network. This is a correlation so far. This is not really proven. We still have to do some, more experiments to be able to really nail that down. But it is an interesting suggestion, I think. Now, I put that in. I know I didn't want to put any electrophysiology in here, but I couldn't resist. And the reason why that is also because I say, what's the motivation? Now, the first time I saw action potential and actually hear them in the old days, you had a speaker connected to that. That really fascinated me and motivated me in the first place to do this kind of science, because this is like, signals from outer space or whatever in our own brain, and this is just amazing. So if you look at we did patch clamp experiments, or actually Sahar, I showed her a picture already, that looked at the electrical activity of these neurons in young animals, and as you see during the day, they're spiking a lot, and during the night, they're less spiking. But in the old animals, this wasn't that clear. So the spike frequency was not really significant in many neurons, was not really significantly different during day and night, which is usually one of the hallmarks of central circadian clocks. So that's and the level of single cells, that is really and also the memory potential doesn't change much, is a problem. Now, if we look at one of the major signals in the SCN, which is important for phase shifting, entrainment, synchronization with the environment, this is calcium, because calcium is mediating the electrical activity to the gene expression, where you basically have the central 24-hour clock generation. So this is usually showed from Chris Colwell and others, modulated that it's higher during the day and lower during the night in young animals. Now, in old animals, it's even reversed. So it's really disturbed that actually you have a larger calcium during the night. And this could be because there's these neurons firing during the night, and you have actually, problem and at the night the calcium is maybe the calcium buffer system may be more sensitive. So this could be that at least the calcium signaling, which is so important for synchronizing and training, is very much disturbed, and that could be a reason why elderly clocks have problems and synchronizing to the outside environment. And not only that, but also the buffer system. And there's very few people that have looked actually at the buffer system. This is actually only an approximation. The first shot at that is to actually see if, we use a very simple measure of potassium deporization induced by high potassium pulse, and looked at the kinetics of the calcium change. And this seems to be slower, which indicates at least some changes in the affinity of the calcium buffers in the cells. And this is also known in other neurons, in aged neurons, to be the case. It could be both ways. It could be an increase in buffers, could be decrease in buffers, but at least the buffers here are so important for intracellular calcium signaling that this is something that may be the basis for this organization. So I already said before that in our hands, the PER2 recordings. So the looking at, and I think there is a movie here. Let's see if that works. Yeah. So this is an SCN, which actually is an animal that produces a fusion protein that shows us, and the glowing bulbs are actually neurons that express a certain clock genes called PER2. And in this case, it's actually a protein, PER2 protein that is equivalent, or the amount of protein is shown by this intensity of bioluminescence. What that allows us and others, there's a lot of people working with that, is to measure the single cell rhythm in gene expression or protein production in SCN. We call that organotypic slices in culture for over several days. We've done that, and we didn't see a lot of difference in the normal LD. This is, again, from Jim Block lab. It's kind of funny that we do some, that he's back in business sometimes. He was a chancellor at UCLA, and is now actually back in the lab. But if you see here that there is, this is the regular activity, this is an aged mouse, and then you see the PER2 rhythms are very similar. There is no difference in peak phase or anything. And we also saw that. What we saw was that we have a difference in the photoperiod. So this was a paper on photoperiod, which I don't want to explain further, but we also saw the same. But the important thing, and that's why I'm showing this, is if you switch the light off and you have the decondition, so if you don't have the light input anymore, the information about the light, this thing is very different. So between aged and young, the PER2 rhythms are really very different in amplitude and phase. So we have a reduced amplitude here, we have a change in phase here. And that tells us that actually the aged clock is stabilized by the regular light input, at least the molecular clock. So it's not completely not affected, and it's only visible when you actually saw that in constant condition. And in the human, which is done by Colin McClung lab, they actually did a time of death experiment where they looked at transcription of genes, and these are just the PER genes to compare that a little bit. And you see in different prefrontal cortex areas that there is some difference in higher baseline, and there is some reduction in amplitude. The PER2, there is a change in phase here. So there are some effects here that you can see. But if you look at the whole data, you see that there's a lot of different phenotypes where you have like base shift or amplitude change. But you also have a loss of rhythmicity in the older, and this is one thing you would usually expect to say, oh, okay, well, getting older, everything, you lose everything. But actually, the gene here, the gene there's other genes that are not rhythmic in younger, and actually gain rhythmicity, and about the same number. So it is suggestive that there is something going on where there may be some kind of reprogramming, although this is, I know, I talked way back to Steve K about this, and he didn't like this word reprogramming, and I understand why, but there is some indication that there's something, trying to rescue the system. And we try to hook onto that and see if we can look at the metabolite. So one of the ways to get at the biochemistry of the cells is to do a metabolomics, and we extracted, we meaning Renate and also Michel van Wegel and myself, we do a lot of extraction from different brain regions and in the periphery also, and we still have actually, I must confess, tissue in the freezer. We didn't have the chance to analyze, and a lot of data already. And then we looked at the different rhythms in metabolites, and what we found, and this is where Lara did her magic, where we could actually look at different, mainly aminos, amines, and we saw that there's, in some cases, there's a different measure, and sometimes there's only rhythm in young, and sometimes only rhythm in old, and this is in different tissues too. So it's very similar that there seems to be that you can actually basically that genes are suddenly, or metabolites are suddenly rhythmic when in the old, which weren't in the young, for instance. So similar to the transcriptomics data. And then what we set out to do is actually to see if we can connect that somehow. Is what is delivered during when the in comparison to the SCN. And so we try to correlate that. And this is also, again, very suggestive, but these are like the correlation lines between the different metabolite oscillations. And you see that actually that in the young, there are many more connections, and of course, between SCN and the output region of the SCN in the hypothalamus epithelium is very tight, whereas there's a lot of connections in this, let's call it metabolomic network, is lost. So we feel like there's a lot going on, which we can of course requires much more research. So what can we do about it? If you look at that, so you wanted to, of course, maybe be more active, or eat at appropriate times and so on. So we divided that in two parts. So there's these lifestyle changes that should enable the clock of the elderly to be more stable, stabilize the clock of the elderly, and the idea is you're exposed to light in the morning, maybe combination with exercise, socializing, which is of course not a given in the elderly a lot of times, regular meal timing and regular rest activity. That will be one of the things, the core things in lifestyle, but not everybody can change the lifestyle. So we actually have other neuroactive drugs, mTOR as one of the, in aging, a big thing a big target for in the metabolism. Melatonin has been used already as a stabilizer and synchronizer. And we actually, at the moment, we have a project together with Paul Höring and Matrei Joshi to look to find new small molecules that actually can enhance the amplitude in the SCN. So what about physical activities? And they don't have to be much actually, but why would physical activity actually increase the? And as you see, this is a very it's all the data, but actually it's still in the making of being published. but you see here from the lab of Joop Meyer, and you see that in the light dark cycle, the MUA activity, the multi-unit activity of the SDN, so electrical activity, is actually in night active animals. During the night, it goes down when it's active. And when you analyze all the times where activity coincides with, so locomotor activity coincides with electrical activity, you actually sum it all up and you see that all of the time that the animal is actually active, the SCN amplitude is decreased here, and here it's increased. So what happened is that an animal that has exercised in the wheel is actually having a larger amplitude than animals that don't exercise, that don't move. So it has a direct effect on the SCN in two ways, that it actually decreases it during the night, and the lack of activity increases it. This may be different in diurnal animals, which we're looking at right now, and actually it's, I think, submitted. So back to the elderly here. So what happens in old mice? So here you see that with exercise, so this is they have a wheel, and there the wheel is locked, and you see that there is some differences in the amplitude already of the behavior. This is all peer passive infrared, so it's a home cage behavior. But if you analyze the quality of the rhythm, they actually drop quite significantly, and even in the young. So in the young, a young animal without a wheel is actually worse than a 1.5 or even 2-year-old animal with a wheel. And luckily, this is reversible. So, if you give them a wheel, then actually the quality increases again. So there is something that, it's some indication that this actually works. And what about drugs? So this is one of the molecules that Jake Chen found in his high throughput screen when he was in Joka Takashi's lab. They call it clock enhancing molecule 3. And we looked at it actually and what we wanted to know in the same prep here is this now every cell increase, or is it a ensemble thing, a synchronization? You already know that if you synchronize it more, the amplitude would increase, or is every cell increased? So you've already shown that the ensemble here in the SCN, this is actually a damaged, it's a mutant SCN here, is delta clock 19, is increased and also. And so what happens in the normal SCN? So Anouk actually was the lead on the study, and we looked at what happens in the Per2 recordings, and you see very nicely that it's actually increased over at least 2 cycles, and this is the summary of it, and it's not due to synchrony. So the synchrony is the same as in the control. So as a conclusion, we see that age affects physiology of single neurons more severely compared to tissue level or behavior. And the age phenotype can be determined by the degree of network disorganization. So the age alteration in GABAergic network, which are mentioned in the EI bounds, may contribute to the changes in phase distribution, but we don't know that yet completely. Finally, age-related changes in temporal order may represent a reprogramming, I'm still going to name that may be beneficial for healthy aging. Now, this completes the data part. I try to keep it short because because I don't want to go too much into detail, given the audience, and I'm very open to questions. But they asked me also to put some personal history here. So this is now where the fun part begins. and so I started in Frankfurt and did my master's, and actually I started with Gerhard Hesmeyer and then went with Günther and Gerhard Fleissner they were really that young then, yeah. And I worked with scorpions, that's already said at that time. And what I did was I looked at temperature-dependent circadian rhythms in scorpions and did some behavioral studies and neurophysiology studies on coupling. So it's already then I looked at coupling and synchronization and so on, yeah. And then, I I did a lot of building at that time and a lot of programming and building. And this is the people that I want to, that are influential to me at that time and that I got to know at that time. And so early on already Jean and got to know Menno and Thiel and Jürgen Aschoff was a big influence, of course. Now then I moved to, as my postdoc I did in University of Virginia with Jean Block. This is when he was a chancellor at UCLA already, so I couldn't find an old picture. And I worked with the sea snail Bulla gouldiana at that point, and I looked at the role of ion channels. So I moved towards patch clamping. Stuart Dreyer at the University of Tallahassee, University of Florida in Tallahassee, taught me patch clamping, and then Sam Schacher from Eric Gendell's lab, he taught me how to do cell culture in mollusk and retina, and Keiko Tominaga from the Inoue lab, she taught me how to do setup organoleptic culture. So you see where I'm getting, you need a lot of help from friends, and you actually, build up a whole group of people that sort of inspire you to do interesting work. So from there, I actually moved to Leipzig to set up my own lab in Leipzig and did my habilitation, whatever that is. And, so this is like qualification to become professor. And this I did with Klaus Schildberger, was from Max Planck Institute in Wiesen, and got a job here as a professor in zoology. Again, I worked with Bulla gouldiana, which is quite a challenge, on land. And I set up a 10,000 liter seawater tank with the help actually from the zoo in Leipzig. And I set up a complete animal physiology teaching lab. So they got you once you have habilitation there. Anyway, and I got to know again a number. Actually, Joke. Yeah, this is the time when I got to know Joke better, and Gabriella, who also was a big influence later. And then money ran out actually, and I got my habilitation. Everything was fine, but they got me, but I couldn't find a job. Was overqualified. So I said I retrain. So Chris invited me to come to his lab to basically start with mammals. At the same time, Nancy, who I got to know from Virginia, she worked on aplesia and egg-laying behavior. So I did a detour and did some neuronal regulation of egg-laying behavior, and by setting up simultaneously recording hormone release, patch clamp, and calcium imaging. That was a lot of fun. I did that with the Swiss Army knife, actually. It was really, really a lot of fun. And I got to know a number of people, and there's only a few of them I'm able to name here. And finally, I moved to where I'm now. Joke invited me to come to Leiden, and this was really an incredible opportunity, and I'm really thankful for that, of course. To kind of finally settle scientifically and also personally. And I continue to work with mice until now. And, there's a lot of projects, including the one I talked today about, and a lot of setups I established, and a lot of great people I got to know. There's no room, as you're already drifting here, no room to name more people. And finally, this was it. And so acknowledgements, of course, the whole group of neurophysiology, and there are some missing for sure. My current project, I work with Paul Höring and Matreya Joshi of chemical biology and drug discovery. Then LCDR, the Laura Heitmann and Ian Snyder's were actually looking at the moment. We're finishing a study where we found the target for Chem3, which was a mystery so far, and Leiden Academic Center, Thomas Hankemeyer and And I'm finished now. I finally made it without the stick being crashed. Thank you very much.